if your mark is 87, right, I would tend to round that down to an 85. Two reasons. Number one, I have to give you a multiple of 5. Number two, if you had an 87 and I gave you a 90, and then at midterm you drop back to your actual mark, which is an 87, I would piss people off because your mark went down. So what I tend to do is I tend to underestimate your mark at midterm. So if you got an 87, I'll give you an 85. And then when it's final report card time, and the mark is actually at 87, it'll go up then from midterm report card. And people like that, which is why I tend to do that. We've been told to, yeah. Because the highest mark we've been asked to give out is a 95. So there's only three. Um, Osmosis cell membrane assignment, the enzymes gizmo, and the biochemistry test. So those are the assignments that we have. In part, I would have liked to have had the cell respiration gizmo in there as well, but not everyone's handed that in. So it wouldn't be fair to have like 3/4 of you get the mark for that assignment, and then the remaining 25% effectively get 0 on it because you haven't finished it yet, right? So I'll probably mark them starting after this weekend, so starting Monday. Or actually, I'll start marking them tomorrow when you guys have your work period. But yeah, they won't end up in your report card. Okay. What else? Is there anything else needs relay for life? No. Okay, so that's basically it. Let's see. That's the other class. Let's go to your class. It will be, yeah. Let's see. Cell respiration. How many of you need to finish it? Okay, I've got 1, 2, 3, 4, 5, 6, 7 people that aren't quite done. One person only has a case study to do. The rest, 1, 2, 3, need to start. And 1, 2, 3, have got maybe 3 questions done, right? So this has to come in because I have to mark it soon. Okay. So I'll work on the ones that have come in so far. Probably on Tuesday, I'm going to start marking these. Like, well, I'm going to mark them no matter what I've got. So if you're only halfway through and you've got, this is out of 55. If you're only halfway through and you got 35 marks, your mark is going to be a 35 out of 55. Yeah, you can start it now if you like. I mean, if you have nothing else to do now, you can start it now. It should be there. I added it this morning, I'm pretty sure. Okay, so I'm going to start with the asynchronous, with the one-on-ones. So let's see. I am starting with whom? Top of the list, Cameron. Kirsten, you're up. Vedant, uh, I'll see you next. Okay, he's not here. Um, the cam stuff. Is there anything I need to share? Oh, so the photosynthesis gizmo. Um, Actually, why don't I create the assignment right now so that you guys have it? So create, reuse a post, gizmo case study, reuse. So the due date for it is going to be a week today, so the 26th. Now that's a drop dead date because it's after the test, and I really need to have those things in so I can mark them. Um, what's it out of? I believe. Let's see. Photosynthesis gizmo is out of a total of 41 marks. You on the 26th, 2017, assign. Okay, so the photosynthesis gizmo, yeah, right here. So due May 26th. Make note of this. Due date is a drop dead date because it shouldn't. You should really. It's about it. Um, once I start marking them, I got. So I'm going to start marking them probably tomorrow because I got to do evidence records today. Um, so while you guys are working on, and the other issue is I've got like 6 people who still haven't handed them in. So I'll start marking them tomorrow. People who haven't handed them in, they're going to get whatever mark they're going to get. Yeah, so that's assigned for next Wednesday, and next Wednesday is a drop dead date. So I'm not going to take them after that Wednesday. Okay, be aware of that. Make sure that I have the same drop dead date for the other class. Few assignments. Okay, what's out of? Okay. Uh, for us, as of today, we were basically finishing off this, which is the cap slides. Just waiting for it to load. Okay, so the other day we discussed what the problem of photorespiration is. Let's bring the whole thing up. Okay, so the issue is that we lose carbons back to CO2. Okay, we reduce the production of photosynthesis because you don't get any energy or food being made. So if we could reduce the amount of photorespiration that's occurring in plants, the plants become 50% more efficient. So there's an incredibly strong selection pressure to evolve a system that doesn't do photorespiration. Okay. Um, the question that I left with you guys with yesterday was, why is it that Rubisco recognizes both this and this as a substrate in its active site? Anyone come up with an answer? Okay, so it does have to do with the shape, right? The active site takes both. So think about when Rubisco evolved. Okay, what were the conditions of the early atmosphere of the Earth like? Yeah. So if this isn't present, does it matter that Rubisco combined either one since this doesn't exist? No. So at the time Rubisco evolved, there was no great selection pressure to differentiate between these two because this one wasn't around. All you had was lots and lots of this. Okay. But today it matters because this is about 21% of our atmosphere. And this is about 0.03 to 0.04% of our atmosphere. Or sorry, the other way around. This is 20%, and this is 0.03 or 4. Okay? So there's a heck of a lot more oxygen than there is O2, CO2, and therefore Rubisco preferentially tends to grab oxygen when it can. Okay? So what we want to do is we want to reduce uh, photorespiration. Okay? So there's basically two ways we can do that. And there are two different plants. Okay, biochemically, the pathway is exactly the same. There is no difference in the biochemistry between these two plant types. What's different is how they actually came up with the same solution. Okay, so C4 plants separate capturing carbon from glucose production. Okay, so different cells capture carbon and make glucose. Okay, the captured carbon is stored as a 4-carbon compound, which is why these are called C4 plants. They also change the enzyme that captures carbon dioxide. It's an enzyme called PEP carboxylase. The leaf structure is also slightly different. Well, actually, quite different. So the anatomy is different. Okay. CAM plants are the other type of method. The biochemistry is exactly the same, but the difference is these guys fix carbon by the time of day. So they fix carbon at night, storing it as a 4-carbon compound, then do photosynthesis during the day. Okay, so let's take a look at both of these. So that's a C4 plant, and that will be a CAM plant. Okay, so CAMs, generally speaking, pineapple, cacti. C4 plants are basically your grasses. Okay, so C4 plants do the following, and I'll put up a Jamboard on this in a minute. Okay, so C4 plants have a different way and a significant better way to capture carbon dioxide. Okay, in the step before Calvin's cycle, they fix the enzyme or capture the carbon using the enzyme PEP carboxylase, right? So they don't use Rubisco to capture carbon, they use PEP carboxylase. Okay, they then store it as a 4-carbon compound. Okay, and it's an adaptation to hot, dry environments. Because in hot, dry environments, what are you going to do with your stomates? Have them open or have them closed? Yeah, because you want to prevent dehydration. Okay, so you have to close the stomates a lot. The anatomy of the leaf is a little different. Okay, so we're talking about things like sugarcane, corn, and other grasses. Does anyone remember from grade 11 what we call things like corn and other grasses in terms of plant type? It's a type of angiosperm. There were two major angiosperms, subtypes. It was related to the kind of seed leaves they had. Okay, what were grasses, sugarcane, things like that? Oh, okay, you missed it. Yeah, Rimka, the monocots, right? So C4 plants are monocots. They only have one seed leaf. So the way you tell them apart, when you look at the leaf, the veins run parallel to each other. So think of grasses, think of celery, think of all the grains that you eat, wheat, oats, barley, they're all monocots. Okay? All right. In order to describe this Jamboard, in order to do this, actually, I am going to launch Jamboard. Now we need a new Jamboard here. So open a jam. Start a new whiteboard. Send. Go full screen. Okay. So I'm doing this on a Chromebook, so it'll be a little bit weird. Share this tab. Okay, so the first one I'm going to basically do is this one here. There's my text box. So this is oops, the C didn't come out. C4 plant. Remember that in a C4 plant, we're separating carbon fixing from um, Kelvin cycle by location. Okay, so what I'm drawing up top here okay, this is something called a mesophyll cell. Okay, right below that is going to be a cell kind of shaped like an oblong trapezoid. Okay, and this is what's called the bundle sheath cell. Okay, and then below that is vascular tissue, xylem and phloem. Okay, now we're going to have carbon dioxide, right? So there's going to be two sets of chloroplasts in here. I'm not going to show the chloroplasts in this diagram, but what I'm about to show you occurs in a chloroplast in the happens as a chloroplast. I do it this way of the mesophyll cell and in the chloroplast of a bundle sheath cell. Okay, so first we're looking at the chloroplast of the mesophyll cell. Okay, so in the chloroplast you're going to have the light reactions going. Okay, so the light reactions are going to generate NADH, pH, and ATP, which will be used by the Calvin cycle later to generate glucose, right? So ATP and NADPH are made up here. Okay, so that's occurring in the chloroplasts here. Okay, so that's occurring up top. Also simultaneously at the same time, CO2 from the atmosphere there we go. From the air is coming into the cell, okay, into the chloroplast. And what's going to actually happen is that a 3-carbon molecule of PEP is going to come along and combine with the CO2 to form oxaloacetate. Because I can. So how many carbons in, excuse me, Joss, how many carbons in PEP? No, think of glycolysis. Remember, same molecule. It's fine. Right, 3 carbons. So there's 3 carbons located there. We're going to add another carbon and go to oxaloacetate. So how many carbons do we have in oxaloacetate? Now your answer, Abby. Okay, so we have 4. Ah, okay, this is why this process is called a C4 plant. Because the first stable process of the carbon capture, the first stable product of the carbon capture produces a 4-carbon molecule. Okay, the enzyme that does this is an enzyme called, why does it keep doing this? PEP carboxylase. Okay, which is pepcase, is the enzyme. Okay, and that enzyme is going to be right here. Okay, so the pepcase enzyme is going to grab CO2 and PEP, combine them into a 4-carbon oxaloacetate. That oxaloacetate will then convert into malate, which is another 4-carbon molecule. Okay, then, not yet, not yet. Then the malate leaves the chloroplast of the mesophyll cell. Then it leaves the mesophyll cell, jumps into the bundle sheath cell, and jumps into a chloroplast of the bundle sheath cell. Okay? So the malate actually exits here and ends up here. Okay? The malate then gets converted to pyruvate. This was 4C. How many carbons in pyruvate? So this is 3C. That means we have to lose a carbon. So a carbon comes out and enters Calvin cycle. Here, Rubisco is going to grab that CO2 and start making sugar. So Rubisco is used, but it's used in the Calvin cycle itself in the mesophyll cell. There is no Rubisco in the mesophyll cell on top. Okay. The pyruvate then leaves the cell and ATP goes to ADP, converting pyruvate into PEP, and the cycle can start again. Yes. So this is basically a CO2 pump. So it captures the carbon dioxide from the air, converts to a 4-carbon product, which will then pump into the bundles, the sheath cell, elevate CO2, and since the light reactions are happening up here, there's no O2 production down here, so Rubisco is not exposed to any O2. Yeah, it takes a small amount of energy, that one single ATP. However, you're going to get a hell of a lot more glucose made, right? Because you're not competing with oxygen anymore. So the light reaction products, malate, have to come down to the bundle sheath cell in order to allow the glucose to be made. Yeah, it's a bit of a filtration process. I think a pump is a better term because the oxygen buildup is happening up here rather than down here. Because up top, right? Where's my Jamboard? Up top here is where the O2 levels are going high. Here, there's no light reaction, so O2 levels stay low. And the CO2 can go into the Calvin cycle and use Rubisco. Okay, that's your basic difference. Okay, so if we go back here and look at this, okay, so this is the dicot plant, C3, right? So you got O2 and CO2 in the airspace, right? Which means you can have photorespiration occurring because oxygen is available. But in a C4 plant, light reactions are occurring up here, driving O2 levels up. But since the Kelvin cycle is sequestered down here and O2 can't get in here, there is no photorespiration going on. That's right, in this system. Okay, so there's a bundle sheath cell. The light reactions are going, generating O2 up here. Okay, pepper carboxylase doesn't react with oxygen, so it completely ignores it. Okay, so PEP plus the carbon dioxide go to oxaloacetate. Okay, the oxaloacetate becomes malate because the stroma of the chloroplast in the mesophyll have none of the enzymes for Calvin cycle in them. The CO2 then gets pumped out. CO2 levels go high here, and Calvin cycle runs with Rubisco. Okay, so pep carboxylase has a higher attraction for CO2 than O2, so it completely ignores CO2. Much better than Rubisco in terms of differentiating between the two. Okay, fixes the CO2 into a 4-carbon oxaloacetate. Okay, then regenerates CO2 in the inner cells for Rubisco and keeps oxygen away from Rubisco. Okay, so that's the basic premise for that. No, you can ferment anything with sugar. Rice should already be C4, I think. Look that up. Oh no, rice is C3. But yeah, you're right. They are trying it from what I'm reading here. Yeah, so it is a C3 plant. So yeah, you can see it here. This was November of 2020. So they took 5 genes from corn or maize and installed it into rice plants. Okay, so they've taken the first steps, really. So they had some small success with it. That's interesting. Okay, so that's a C4 plant. Okay, so you can see here's the comparative anatomy in a C3 versus C4. So this is your C3. This is your typical plant, right? And this is a C4 plant. You see the difference in structure, right? So there's air gaps all over the place here, whereas here it's tightly filled, right? Much smaller air gaps. That helps keep the oxygen away as well. Also, the light reactions, remember, are only occurring here. So these will build up O2, but since they use pepcase, they're not going to react. Pepcase isn't going to react to the oxygen building up here. Then you go to the bundled sheath cell here to make your sugars. Okay, so for this particular system, the monocots, it's all about location, location, location. Okay, the other possibility is the CAM plant. Okay, and in a CAM plant, like I said, the biochemistry is exactly the same. Okay, the difference is in time of day as to when carbon fixing is going to occur. That's the basic difference. Okay, let's do this note first. So when we talk about CAM plants, CAM stands for crustaceous acid metabolism. Okay, it's an adaptation to dry, hot climates. Okay, so we're separating carbon fixing by Calvin cycle by time. The biochemistry is exactly the same. Okay, we're going to close the stomates during the day because that's when we want to prevent water loss, and we open stomates at night because that's when we want to capture the oxygen. Okay, so at night you open the stomates and fix the carbon, and during the day you release the CO2 from the 4 carbon acids to the Calvin cycle. So we're talking about things like succulents, like aloe vera, some cacti, and some pineapples. Okay. Now, in terms of the metabolic pathway that we're talking about, here's what's basically going on. Okay. So first off, this is at nighttime. Whoop. Okay. During nighttime, we're going to have CO2 in the air coming into the plant, okay, because the stomates are open. Okay. So the CO2 comes in to the plant leaf. You're going to have PEP sitting here waiting. Okay. So it's going to combine using pepcase. Okay. So these two will combine 3 carbon plus 1 carbon to make oxaloacetate.
Oh, you aren't presenting.
Yeah, you should be logged in though.
Okay, now you're presenting.
Okay. So we've got oxaloacetate. Okay. That oxaloacetate now, just like last time, is going to get converted into malate. Okay. And now the malate just sits there. It doesn't do anything. Okay. Now what we're going to have, okay, this is going to be daytime. So stomates close. Okay. So the malate, which has just been building up, comes here. And it's going to do the exact same thing as before. it's going to turn into a pyruvate and give off a CO2. That CO2 is going to go into the Calvin cycle. Okay? The pyruvate is going to get acted on by ATP, converting it to ADP, and it's going to become PEP. The PEP now builds up and waits until nighttime when the stomate's open again and the system can restart. Okay? So the chemistry is exactly the same between here and here, right? The components are exactly the same. What's different, this is by location, mesosphil versus bundle sheath. This is by time, nighttime versus daytime. Okay? In both cases, this part is basically acting as a pump to increase CO2 levels and keep oxygen levels low such that the Calvin cycle is not going to, it's going to, the Rubisco isn't going to use the oxygen. Okay? So in this case, it's all about the timing. Okay? So these are pineapples, some cacti, and some succulents. Okay? So if you look at this, right, C4 plants separate the two steps of carbon fixation and glucose production anatomically in two different cells. C3 plants separate the two steps temporally by time, night versus day. But the steps are exactly the same between the two processes. We talked about why the C3 problem exists. Okay. It's because it's just evolutionary baggage, which we discussed earlier. Okay, that's it. We are done this unit. So, what time is it? 1:40. Take a 10-minute break, and then when you get back, so I'll see you at what, 1:51. Start working on the gizmo. So you now have all the information you need to do the gizmo. So see you in 10. So, work on the gizmo, and I guess I am doing report cards.